Hey everyone, and welcome back to the deep dive. Today we're going to be looking at some really interesting research on oral cancer, specifically how stem cells can go from completely harmless to fueling a tumor. It's all in this study that was just published in Nature Communications. And you know what? The thing that really caught my eye is how fast this whole process can happen. We're not talking years here. We're talking weeks.
Yeah, it is really remarkable how quickly things can change at that cellular level. And this study, you're right, it really highlights how important it is to understand those early stages of how cancer develops.
For sure. So to get us all on the same page, we know that things like tobacco and alcohol are major risk factors for oral cancer. But did you know that something like 30% of oral cancer cases are linked to HPV?
That's right, the human papillomavirus. And this study really focuses in on HPV-positive oral cancer, looking at those early molecular events that, we don't always see.
Okay, so HPV is involved, but it seems like the real star of the show here is this protein called YAP. Can you tell us a little bit more about YAP and why it's so important in this whole process?
Well, YAP stands for yes-associated protein, and normally it's involved in healthy cell growth. But when it's not regulated properly, it can actually become kind of a troublemaker. You can think of it like a car with a broken braking system, right? Things can get out of control really quickly. And the braking system here is a cellular pathway called the Hippo pathway. So when the Hippo pathway isn't working properly, YAP can basically run wild. And this can lead to uncontrolled cell growth and eventually cancer.
Wow, so YAP is like this little speed demon in our cells if it's not kept in check. So how did the researchers actually figure this all out? Did they watch cells under a microscope or something?
Well, not exactly. They were pretty clever. They actually used a mouse model where they could activate YAP, the HPV oncoproteins E6 and E7, or they could activate both. And what's really striking is that the mice that had both YAP and those HPV proteins activated, we call them the EY mice, they developed visible tumors in just 2 weeks.
2 weeks? That's kind of scary. What's going on inside the cells to make them change so quickly?
Right. Well, to figure that out, the researchers used this technique called RNA sequencing, which is basically like taking a snapshot of all the genes that are active in a cell at a certain time. And what they saw in the EY mice was really interesting. It was almost like gene expression chaos. They found over 2,000 genes that were behaving differently compared to the control mice. So it was a huge shift in the cellular activity. And a lot of these genes were related to cell growth, the development of that lining of the mouth, and inflammation, which are all hallmarks of cancer.
Oh, wow. Okay, yeah, that makes sense. So the genes are going haywire, but how does that actually turn a normal cell into a cancerous one? What's the connection?
So remember that mTOR pathway we talked about earlier, the one that's often involved in cancer? Well, it seems like that pathway is really involved in this whole cascade of changes, too. And on top of that, the researchers also noticed what we call epigenetic reprogramming. Think of it like this.
Yeah.
Your DNA is like the hardware of your cells, and the genes are the software. Epigenetics is kind of like tweaking the software, making some genes easier or harder to run. And in this case, the epigenetic changes they saw were actually making those genes that are involved in cancer development more accessible. So that was kind of like adding fuel to the fire.
So it's like the cancer is hijacking the cell's operating system. That's pretty sneaky. But they didn't just stop there, did they? I think I read that they were able to look even closer, at the level of individual cells.
Yeah, you're right. They wanted to go deeper. So they used this really powerful tool called single-cell RNA sequencing. And this is where things get really interesting. They discovered that in the EY mice, there was this cluster of cells that looked a lot like what we call tumor-initiating cells. And those are the cells that actually drive the growth and spread of the tumor.
So it's not just that all the cells are going bad. There's this specific group of troublemakers. And these troublemakers, they were once just regular stem cells, right?
Exactly. They were just doing their job, maintaining the lining of the mouth. But because of the YAP activation and those HPV proteins, they got reprogrammed into a more dangerous state, one that's more likely to invade other tissues. It's like they flipped a switch in their programming, going from maintain and repair to grow and invade.
Wow, that is wild. So we have these stem cells that have gone rogue, turned into these tumor-initiating cells. But they weren't acting alone, were they? There's more to the story, right?
You're right, there is. It turns out that these tumor-initiating cells were actually calling for backup.
Backup,
Wow.
like more stem cells.
Nope. They were calling in immune cells, specifically a type of immune cell called granulocytes. So the researchers found that the tumor-initiating cells were sending out these signals, almost like little distress flares, that were attracting these granulocytes to the tumor.
Wait a minute, isn't the immune system supposed to fight cancer? Like, isn't that its whole job? Why would these granulocytes be hanging out with the tumor cells?
I know, it's kind of weird, right? But that's what makes this so interesting. These granulocytes were actually producing an enzyme called collagenase. And collagen, if you think about it, is like the scaffolding that holds all of our tissues together. And what does collagenase do? It breaks down that scaffolding.
Oh, no, so it's like the tumor is creating a path for itself to escape.
Exactly. By breaking down that collagen, the tumor cells can more easily invade the surrounding tissues. It's almost like the tumor is manipulating the immune system to do its dirty work.
So we have these stem cells going rogue, becoming these tumor-initiating cells, and then they're tricking the immune system into helping them spread. It's like some kind of cellular conspiracy.
It really does highlight how complex cancer can be. It's not just about one gene or one cell type going wrong. It's about all of these different players and all of these different signals that are going back and forth. It's a whole network of interactions.
Yeah, it's a lot to keep track of. But I think we've done a good job of breaking it down so far. We've identified this potential culprit, YAP, and we've seen how it can team up with HPV to really create a recipe for disaster. But now, what can we actually do about it?
Well, that's a great question, and it's one that researchers are working on right now. One really promising avenue is to develop therapies that target YAP directly, basically to try to cut off the head of the snake.
Okay, that makes sense. But would that be a treatment just for oral cancer? Or could it potentially be used for other types of cancer, too?
Well, the really exciting thing is that YAP seems to be involved in other types of cancer, not just HPV-positive oral cancer. So if researchers can figure out how to effectively inhibit YAP, it could potentially have a much wider impact on cancer treatment in general. Imagine if we had a drug that could actually tackle lots of different types of cancer all at once. Of course, there are challenges. We need to make sure that these inhibitors are safe and that they're actually effective. And we need to make sure that they don't interfere with the normal functions of YAP in healthy cells.
Right, so it's not as simple as just completely shutting YAP down. We need to be a bit more strategic. But yeah, if we could figure that out, it could be huge. What about preventing cancer in the first place? Is there anything we can do to prevent this whole cellular coup from happening?
Well, one of the most important things is early detection. The earlier we can catch these changes, the better our chances are of stopping the tumor before it really takes hold. And of course, reducing exposure to those things we know are linked to cancer, like tobacco and alcohol, is also really important.
Yeah, those are the classics. But what about HPV? Isn't there a vaccine for that?
Yes. The HPV vaccine is a really powerful tool we have for preventing HPV-related cancers, including oral cancer. It's safe and it's effective, and it's something that everyone should consider, regardless of their age or gender.
So we have early detection, healthy lifestyle choices, and vaccination. Those are our best weapons in the fight against oral cancer. But before we move on, was there anything else in this study that stood out to you? Anything that maybe our listeners should pay particular attention to?
You know, the thing that really got me was how quickly those tumors developed in those EY mice. 2 weeks. It really just emphasizes how quickly cancer can progress, sometimes much faster than we think.
Yeah, that is really scary to think about. It really drives home the point that understanding those early stages of cancer is crucial.
Absolutely. If we can identify those early warning signs and intervene quickly, we have a much better chance of stopping the disease in its tracks.
So it's not just about finding a cure, it's about preventing cancer from even getting started. All right, well, we've covered a lot of ground here. We've talked about rogue stem cells, potential new targets for therapy, and the importance of early detection. But before we move on, I think it's worth taking a closer look at some of the tools that researchers are using to make these discoveries. Like, for example, RNA sequencing and that single-cell RNA sequencing. Can you tell us a bit more about those? What do those techniques allow researchers to do that they couldn't do before?
Sure. So before we had RNA sequencing, scientists had to study gene expression in bulk, basically looking at the average activity of thousands or even millions of cells all at once. It's kind of like listening to an orchestra play, but you can only hear the whole thing at once. You get the general idea, but you can't really pick out the individual instruments.
So you get the overall sound, but you miss all the detail.
Yeah, exactly. But with RNA sequencing, we can actually see the individual instruments, each gene, and we can see how it's contributing to the overall composition. We can see which genes are turned on, which ones are turned off, and how much of each gene is being expressed.
Wow, so it's like going from a blurry picture to a high-definition one.
Exactly. And single-cell RNA sequencing takes it a step further. Now, instead of listening to the whole orchestra at once, we can focus on a single instrument, a single cell. In this study, that's how they were able to identify those tumor-initiating cells. They were able to zero in on those specific cells, even though there were all these other cell types present.
Wow, that is amazing. It's like we've gone from looking at a crowd to being able to pick out individual faces.
Yeah, that's a great way to put it. And this level of detail is really important because it allows us to understand how different cells communicate with each other, how they influence each other, and how they ultimately contribute to tumor growth.
It's like we're getting this inside view of how cancer actually develops at the cellular level.
It is. And I think it's important to remember that there's still so much we don't know. Every new discovery just opens up more questions, more avenues for research.
Well, on that note, I think it's time to take a quick break and come back to talk more about the implications of this research. We'll explore the future of YAP-targeted therapies, the importance of early detection, and what other surprises might be lurking in the microscopic world inside us. Stay tuned.
Yeah, it's really fascinating stuff, and there is still so much that we don't know.
All right, so let's get back into it. Before the break, we were talking about the potential of developing therapies that could directly target YAP. This seems super promising, but I'm curious to know, what are some of the challenges involved? What are those hurdles that researchers have to overcome to make this a reality?
Well, one of the biggest challenges with targeting YAP is specificity. You see, YAP actually plays a really important role in healthy cells too, especially during development and when tissues are repairing themselves. So we can't just go around blocking YAP everywhere in the body because that would cause a lot of problems. We need to find a way to target it very specifically to the cancer cells and leave the healthy cells alone.
Right, so it's kind of like trying to disarm a bomb without accidentally setting off any other explosions.
Yeah, exactly. And another challenge is delivery. Like, how do we get those YAP inhibitors just to those tumor cells? If they're floating around everywhere in the body, they could end up causing some pretty nasty side effects. So scientists are looking at different ways to kind of sneak those inhibitors in. Things like using nanoparticles or special antibodies that can deliver the drug right to the tumor.
It sounds like a really delicate balancing act. But if we can figure it out, it has the potential to be huge, right?
Absolutely. If we can develop safe and effective YAP inhibitors, it could be a game changer for cancer treatment. Not just for oral cancer, but potentially for lots of other types of cancer as well.
That is really exciting to think about. But while we're waiting for those breakthroughs to happen, what can people do right now to protect themselves? We've talked about early detection and making healthy lifestyle choices, but is there anything else?
Yeah, those are all super important. But there's another thing that people often overlook, and that's oral health. Going to the dentist regularly for checkups is not just about preventing cavities, it's also about catching any early signs of oral cancer. Dentists are often the first ones to spot anything suspicious, like lesions or changes in the mouth that you might not even notice yourself.
So it's about being proactive and staying on top of those dental appointments. And for people who are eligible, the HPV vaccine is another great way to protect yourself.
Definitely. The HPV vaccine is really effective, and it's something everyone should consider. All right, so we've talked about early detection, healthy habits, and vaccination. Those are our best tools for fighting oral cancer. But let's zoom out for a second and think about the bigger picture. This study looked at HPV-positive oral cancer, but we know that YAP is also involved in other types of cancer. So what does this research mean for cancer treatment in general?
That's a great question. I mean, it seems like we're moving away from that one-size-fits-all approach to cancer treatment.
Exactly. Cancer isn't just one disease, it's a whole group of diseases, and they all have their own quirks. So treating all cancers the same way just doesn't make sense.
So instead of just looking at where the cancer is, like breast cancer or lung cancer, we need to start looking at what's actually driving it at a molecular level.
Exactly. And that's where research like this comes in. By understanding those specific molecular pathways, we can develop more targeted and effective therapies. It's all about tailoring the treatment to the individual patient, which is what we call precision medicine.
Oh, right. Precision medicine, like creating a custom-made suit instead of trying to fit everyone into the same off-the-rack size.
I like that analogy. And this study is a perfect example of how basic science research can pave the way for those more personalized treatments.
It's amazing to think that studying something as small as a protein can lead to such huge advances in cancer treatment.
It really is.
Yeah.
And it speaks to the power of scientific curiosity and our constant drive to understand how life works and use that knowledge to improve human health.
Couldn't have said it better myself. And as we continue to delve deeper into that molecular world, I'm sure we're going to uncover even more secrets that could revolutionize how we approach cancer care.
I completely agree. The future of cancer treatment is bright, and studies like this one are lighting the way.
Okay, so we've talked about the cellular level and the big picture of cancer treatment. Before we wrap up this deep dive, is there anything else that you think really stood out in this research? Something that our listeners should keep in mind?
Well, one of the things that I found really impressive was how the researchers used that single-cell RNA sequencing technique to zero in on those tumor-initiating cells. That technology has really been a game changer for cancer research. It allows us to study cancer in a whole new way.
It's like we went from looking at a blurry photograph to having a high-resolution image where we can see every little detail.
Exactly. And that level of detail is really important because it helps us understand how those different cells are interacting and how they're contributing to tumor growth.
And the more we understand about those interactions, the better we can target them with new therapies.
Exactly. By understanding the molecular profiles of those individual cells, we can develop drugs that specifically target those bad actors while leaving the healthy cells alone. It's a much more precise approach.
It's like using a scalpel instead of a sledgehammer.
That's a great way to put it. And I think that's really the future of cancer treatment, moving towards those more personalized and precise therapies.
It's a really exciting time to be following this field. It seems like there are new discoveries happening all the time.
It really is, and I have a feeling that we'll be doing a lot more deep dives into this topic in the future.
Yeah.
There's still so much to learn.
Well, on that note, let's take another quick break. When we come back, we'll wrap up our discussion and explore some of the thought-provoking questions that this research raises about the future of cancer treatment. Stay tuned.
Welcome back to the Deep Dive, everyone. We're here wrapping up our exploration of stem cells and oral cancer. Man, it's been quite a journey, hasn't it? We've seen how these seemingly harmless stem cells can turn rogue and start fueling tumor growth. We learned about this super important protein, YAP, and of course we talked about the importance of early detection and targeted therapies. But as with any deep dive, I feel like we've come up with more questions than answers. I think that's the really exciting thing about science. Every time we learn something new, it just opens up a whole new world of possibilities and mysteries to solve. And this research is a perfect example of that. We're getting a better understanding of all the complex processes that happen inside our cells when cancer develops. But it also makes you think about, just the nature of our cells and how they can be both good and bad.
Yeah, it's like this cellular version of yin and yang. This potential for healing and growth, but also this potential for destruction and disease.
I love that analogy. It really captures the duality of our biology. We're always walking this fine line between health and disease, and ultimately it's our cells that decide which way we go.
So tell me, what really stood out to you in this research? What was your biggest aha moment?
Honestly, it was just how quickly those tumors developed in the mice. Like we've been saying, 2 weeks to go from healthy to having visible tumors. It just shows that cancer can be so much faster and more aggressive than we often think.
Yeah, that's a pretty sobering thought. It definitely highlights the importance of catching things early.
Right. And it also makes you realize just how powerful our own cells can be. If these stem cells can be hijacked and turned into tumor-initiating cells, it makes you wonder what other secrets are hidden inside us, just waiting to be discovered.
Okay, so speaking of hijacking, earlier we were talking about the possibility of actually reprogramming those tumor-initiating cells, turning them back into normal, healthy cells. That sounds kind of crazy, but it's something scientists are actually working on, right?
Oh yeah, definitely. The field of cellular reprogramming is exploding right now, and scientists are making some amazing progress in transforming one type of cell into another. It's still early days, but if we can figure out how to do that with cancer cells, it would be incredible. We could essentially undo the damage that the cancer has done.
Wow, so instead of killing cancer cells, we could actually heal them, restore them to their original programming. It would be a total paradigm shift in medicine.
Absolutely. And research like this, which focuses on understanding those molecular mechanisms behind cancer, is laying the foundation for those kinds of breakthroughs.
It gives you hope that even though cancer is such a formidable opponent, we are making progress. With every new discovery, we get a little bit closer to finding a cure.
I couldn't agree more. And I think this research really drives home the message that even though cancer can be really scary, it's not an unbeatable foe. There's always hope, and we're constantly learning more and getting better at fighting it.
Well said. Well, listeners, we've reached the end of our deep dive into the fascinating world of stem cells and oral cancer. I hope you found it as informative and thought-provoking as we did.
We've covered a lot of ground today, from the microscopic details of how cancer develops to the big picture of treatment and prevention. But most importantly, I hope we've inspired you to keep learning and exploring the amazing world of science.
And if you want to learn even more about the research we talked about today, be sure to check out the full study in Nature Communications. We'll have a link to it in the show notes.
And as always, stay curious, keep asking questions, and keep those minds open to new possibilities.
Thanks for joining us on the Deep Dive. Until next time, stay curious and keep exploring.